Hello students, welcome to Showed classes. So in this lecture we will be starting with IIT JAM Biotechnology. So the topic of today will be metabolism. What is metabolism? What is the definition of metabolism and how these processes going on? So this is the first introductory part we should learn before we get into some metabolic pathway. So what is metabolism? Metabolism is some catalytic reaction that occur through a series of enzyme catalyzed reaction, enzyme catalyzed reaction, and forming some product reaction. Forming some product. These products can be intermediate products. These intermediate products are mainly, we call it as metabolites, intermediary metabolites. And after that there are some products that are forming intermediately, but they are actually used for many huge role in the living system or living cells. So these products are we calling it as metabolites. And the process of these, all of the chemical transformation is called as metabolism. Now this reaction, this metabolism, this reaction occurs to obtain some chemical energy, or to convert nutrient to some biomolecule or the characteristics molecule, convert nutrient to some molecules for the growth and the metabolism, or the growth and development. Now the third one is synthesize and degrade biomolecule. Degrade or to synthesize biomolecule to require some cellular function to happen. For some cellular function. So these are the main function or the main significance for the metabolism or metabolic reaction. Now we can subdivide these metabolic reaction into two classes. Metabolic reaction that can be of two types. First one is the catabolism. This is the first one. The second one is anabolism. Now the major difference of the first, let me tell you what is the similarity of the catabolism and anabolism. From a complex molecule, the only idea is to make simple biomolecule for the development, growth, and for other reaction, biochemical reaction. But in case of catabolism, the bigger molecule, the complex molecule, are degrading into simple forms, And in case of anabolism, it is the synthesis phase when in which the larger and more complex molecules are synthesized from small molecules. There are small molecules and they are giving rise to a molecule, a more complex molecule, so that can process this reaction, and these reactions are undergoing as they require the input of energy. They require ATP, NADH, NAD, FADH2. So they are requiring the power of these. The pathway that can be either anabolic or the catabolic. Okay, this metabolic reaction that can be happen by this catabolic pathway or by the anabolic pathway. And the metabolic pathway are regulated from, are being carried out from outside by different ways, such as the availability of substrate. The availability of substrate. The second is allosteric regulation. Allosteric regulation by the metabolic intermediate or coenzyme. Third one is by growth factor and hormones. Hormones and growth factor. Then there are extracellular signals, intercellular signals that are giving rise to these metabolic reactions. Now, this was the introductory part for the metabolism. Why metabolism are happening? What is metabolism? And what are the procedures? Now some of the metabolic reaction that we have to study, and we will be studying with the easiest one, that is the glycolysis. But remember one thing, this glycolysis is the basic pathway for the metabolism. But you have to remember one thing, this is a very important pathway of metabolic pathway. Okay, so we have to study it very carefully. Now by the term glycolysis, what you can understand, that is it's forming glucose, no, that is it's lysis of the glucose. There is a lysis part and the glyco part. Means glyco means the sweet or the glycogen, glyco means sugar, and lysis means breaking. So from here we can say there is a lysis of glucose. Okay, so this is the thing. Now it is also known as Embden-Meyerhof pathway. It's also called as Embden-Meyerhof pathway, as it was discovered by Embden and Meyerhof in 1930. Now glycolysis is the sequence of enzyme catalyzed reaction that metabolize one molecule of glucose to two molecules of pyruvate. The basic is glucose, one molecule of glucose to two molecules of pyruvate. So with the net production of two molecules of ATP. Two molecules of ATPs. So two molecules of ATPs are forming and two molecules of pyruvate. So as it is the major pathway of glucose metabolism that occur anaerobically, means there is no needs of air or the and these occurs in the cytosol of the cell. Cytosol of the cell. And one more thing that is about the glycolysis, it is a 10-step process. 10 steps are there. 10-step process in which 6 carbon glucose break down into the different molecules, and then the 3 carbon pyruvate, 2 molecules of 3 carbon pyruvate that forms. These 10 steps are divided into 2 phases. First is preparatory phase. Preparatory phase. Now what is preparatory phase? And the another one is payoff phase. Payoff phase. In preparatory phase, that requires the ATP. Okay, there is a gain of ATP in the payoff phase, and it requires the ATP, means they need ATP from outside. And in payoff phase, there is the production of ATP. That is the difference of preparatory phase and payoff phase. So let's start with the phases that is conducting in metabolism of glycolysis. So the step 1 of the preparatory phase of glycolysis includes glucose to form a glucose 6-phosphate. Glucose 6-phosphate. And the enzyme that is helping this formation or conversion is hexokinase. Hexokinase. And with the help of ATP to ADP. ATP to ADP. So this is the conversion of glucose to glucose 6-phosphate. That is the phosphorylation of the glucose in this pathway. Now the formation of glucose 6-phosphate to the another isomer, that is the isomerization reaction. This is the one-way reaction, that is why this is the single arrow, and here this is the isomerization reaction, means the glucose 6-phosphate is the isomer of another molecule that is called fructose 6-phosphate, that is the ketose form. That is the fructose 6-phosphate. And the glucose 6-phosphate is the aldose form of a molecule. This is the aldose form and this is the ketose form. So here the enzyme that is helping this isomerization is phosphoglucose isomerase. Phosphoglucose isomerase. Here the rearrangement of the carbonyl and the hydroxyl group at C1 and the C2 is necessary for the next two steps, and the another molecule of ATP is consumed here. So here at this level, in the formation of these fructose 6-phosphate to form fructose 1,6-bisphosphate, fructose 1,6-bisphosphate is required, that is PFK1. I have that should be in the PFK1, that is phosphofructose isomerase or kinase, that is phosphofructose kinase 1 is required here to form fructose 1,6 bisphosphate. So here this is okay, another ATP will be consumed here, so that is first writing this ATP to ADP, and that will form fructose 1,6 bisphosphate. Fructose 1,6 bisphosphate. Okay, so from the first glucose is first converted to the glucose 6-phosphate, and then there is a isomerization reaction, that is glucose 6-phosphate to the fructose 6-phosphate, and then another molecule of ATP is consumed, and here the enzyme is phosphofructose kinase 1, and that forming fructose 1,6 bisphosphate. So fructose 1,6 bisphosphate, there actually what happens, it also transfer a phosphoryl group from the ATP irreversibly. Okay, so this is okay, this will be a one-way arrow. This is the one-way arrow because this is happening irreversibly from fructose 6-phosphate to fructose 1,6 bisphosphate. Now after fructose 1,6 bisphosphate, there is two ways that are forming. First is here, that is forming the dihydroxyacetone phosphate or DHAP, DHAP, dihydroxyacetone phosphate. And the another one that will be forming from this is also in this formation, that is reversible formation, and from here that will be forming this. Okay, so this will forming glyceraldehyde 3-phosphate, glyceraldehyde 3-phosphate, that is GAP. GAP. Now from glyceraldehyde 3-phosphate to form fructose 1,6 bisphosphate to DHAP and GAP, this dihydroxyacetone phosphate and the glyceraldehyde 3-phosphate, here the enzyme aldolase is we are using, aldolase. Okay, so this is the fourth step. Here this one is the third step. This is the third step. This is the second step, and this hexokinase is the first step. So here ultimately we have these. Okay, so glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Now from these there is a interchange, like they can be interchanged between them by the enzyme, because this is also another isomerization reaction, triose phosphate isomerase. Triose phosphate isomerase. Okay, so this is the interchange isomerization reaction. So this is the fifth step. Actually this is not the fifth step, okay, but here the another reaction is happening, that's why we call it as fifth step. So these are all the preparatory phase where these ATP is consumed to form two molecules of ATPs are consumed here. So here after this there will be formation of the ATPs, okay. And another thing, this glyceraldehyde 3 phosphate gap, that is forming in two molecules of gap glyceraldehyde 3 phosphate is forming. So from here the starting of the payoff phase, okay. So from glyceraldehyde 3 phosphate there is another. So from here glyceraldehyde 3 phosphate that will be forming 1,3 phosphoglycerate. So this 1,3-bisphosphoglycerate, this is actually this is, I should have written in this pen. Well, this is this conversion is by the enzyme glyceraldehyde 3-phosphate dehydrogenase. 3-phosphate dehydrogenase. Okay, this is the enzyme here we are using, and there are some also other molecules like 2-phosphate molecules are using here, and also 2 molecules of NAD plus consumed here to form 2 NADH and H plus. So from here this is the sixth reaction and the first reaction of the payoff phase. So the next one is from 1,3-bisphosphoglycerate to 3-phosphoglycerate there is the formation of the 2-phosphoglycerate. 2-phosphoglycerate. This 2-phosphoglycerate, here the enzyme is using phosphoglycerate kinase, because this is the first ATP there is coming or forming, okay. So this is 2 ADP as consuming to form 2 molecules of ATP, and the enzyme is phosphoglycerate kinase. So this is the 8th reaction. This was the 7th reaction. Now after 2-phosphoglycerate, from 2-phosphoglycerate we are forming the 9th reaction, that is phosphopyruvate. Phosphoenol pyruvate. So phosphoenol pyruvate, that is two H2O molecules, water molecules are coming out from these reaction, and this is the ninth one. This is the ninth reaction, and here the enolase enzyme is using for this conversion. Enolase. Now from phosphoenol pyruvate there is an irreversible reaction, that is the second ATP is forming from two molecules of ADP. Two molecules of ATP is forming, and pyruvate kinase is the enzyme kinase to form pyruvate. So this is the 10th reaction, the last reaction of the payoff phase, and this is all about the glycolysis, that is the conversion of glucose, that is the dissociation of glucose to form undergo a metabolic reaction to form the pyruvate as the end product. So thank you for watching this video.